Okay, hello AP Biology. So today what we're going to do is we're going to do a follow-up session here on the activity that we did yesterday with an action potential in the nerve cell. All right, so as we move through here, my expectation is that you should be thinking about what you did in the activity yesterday. You also can feel free to pause this video along the way and go back and revisit what is going on at each section. When we are done with the video, you will go back and go through the activity again and start to add a bit more detail to it so that you can really see what's going on with the action potentials here. Keep in mind, I know there's a lot going on. There's some big words. It's sort of complicated, so you may have to watch this video more than once. Okay, so the first thing we're going to do is we're going to talk about the resting neuron. Then we're going to talk about what does it mean to have an action potential. This is really what is making these nerve cells send their signal from one end, from the dendrite end, down through the axon end. Okay, what is it? What does that message look like? Well, in a nerve cell, it is a lot of electrical stimulation that travels down. And as we talked about before, it's a domino effect where once we take the first domino and we get enough energy to push that, or enough stimulation to push that first domino over, then we start to see a series of reactions that take place down the line of those dominoes as one domino knocking over the next, knocking over the next, knocking over the next, until the last one falls on a ball and it ends up in a hoop or something like that. Okay, so a little bit of review here. That's all of your neurons or your cells are surrounded by a solution, and those solutions have different ions that are dissolved in it. The inside of the cell also is full of cytoplasm, which is a solution, and it has quite a bit of ions in it as well as other structures. Okay, the ones that we're really concerned about with the nerve cell are the amino acids that are found within the nerve cell itself, and those amino acids eventually will go on to form those neurotransmitters, or the hormones of the brain, that go on to go through that gap, the synaptic gap. Okay, so the ions that we're really concerned about at this point are the sodium ions and the potassium ions. If you remember the salty bananas, salty is on the extracellular portion of the cell, and the bananas are intracellular. Sodium is from the salt, and potassium we get from those awesome bananas. Okay, so these cells live in a sea of ions. Okay, anions are your negatively charged ions, and they are in high concentration within the cell, specifically the chlorine ions, as well as the amino acids. Okay, your cation ions, oops, there we go. The cation ions are your positively charged ions, and those are your sodium ones that are in higher concentration on the outside of the cell, although the potassium ions are positively charged as well. They're just not, they're being outnumbered by those anions, or those negative anions inside the cell. Okay, so more concentrated extracellularly, the positive charges, and those are your potassiums. All right, in order for both of the sodium and the potassiums to travel through that membrane, because they are polar, they are charged ions, they are not going to be able to get through that phospholipid bilayer, and so they need to travel through channels. And in this particular situation, the type of channel that we're talking about here are going to be voltage-gated channel proteins. And they are called voltage-gated proteins because in order for them to open up and allow their substance to travel through the protein, or travel through the channel, it is going to be an electrical gradient that is going to cause these channels to open up. And we'll see that as we start to move through here. Okay, cells have voltage. They are opposite charges on the opposite sides of the membrane. The inside of the membrane is more negative than is the outside of the cell. And we can go through and we can record that information with a voltage meter, and we find that the inside of the cell is minus 70 millivolts. Now that doesn't mean it's its actual charge. It just means that it's 70 millivolts less than the outside of the cell when we compare them. Okay, so the outside of the cell might have a positive charge as 600 millivolts, and so the inside might be 530 millivolts. Okay, but because it's less, we say that it is negative. Okay, So it's charged, so therefore the membrane is polarized. Okay, keep that word in mind because we're going to be using the word polarized as we go through and we talk about what's happening with these action potentials. Okay, here is our gradient. Okay, and because we have these built up of these anions and these cations inside and out, it's going to store this energy. That is our potential energy, and we're going to see this travel down the axon of our cells. All right, so the first thing that happens when we start to get a nerve impulse or to cause the nerve to actually work is there has to be a stimulus that is received. Okay. Once the nerve is stimulated, this is going to cause the sodium channels to open up. Okay. We always talk about things in terms of sodium and potassium. So one way to remember this is that we always work with sodium first, and then we go to potassium. Okay. So the sodium channels are going to open up, and because we have two gradients working here, sodium is going to diffuse into the cell. It is going to follow its concentration gradient. So we have a lot of sodium outside, not so much inside, and so sodium moves from a high concentration to a low concentration. Down the concentration gradient we go. Hi ho, hi ho. All right. So not only do we have the concentration gradient here, but we also have an electrochemical gradient or electrical gradient because it's positive on the outside, negative on the inside, and opposite charges attract. So sodium is going to be attracted to that negative inside. Okay, so when these sodium channels open up, this is our first domino going down. Okay. Again, the stimulus is applied, so it's coming from all of these different directions, and we have to be able to narrow it down. All right, the stimulus causes the sodium gates to open. There they go. Sodium is going to move into the cell. There it goes. And once the gates have been opened, they're really open a very short amount of time, about 1 millisecond. And so if you remember from the activity yesterday, we spent about 10 seconds moving sodium into the cell. Okay. Action potentials. This is what really is going on. This is describing the change in the charges between the inside and the outside of the cell. Because the sodium is rushing into the cell, we are now taking a lot of positive charges and adding them to the inside of the cell. So the inside of the cell then becomes less and less negative, or it starts to move towards the positive charge, which means that we are becoming less and less charged. Okay, so what we see then is that this membrane is starting to become depolarized or unpolarized, going from the negative up towards the positive. Okay, and this is all due because the sodium channels are open and sodium is moving into the cell. All right, so then what we see is that once those once the potassium ions move into the cell, then what we see is that the charges will actually swap place, and the outside of the cell then will be more negative with respect to the inside of the cell, and we'll see this in just a minute. Okay, keep in mind we're only talking about sodium channels. The potassium channels are still sitting there doing absolutely nothing. All right, so this is what our graph looks like of an action potential. When a stimulus is applied, a resting neuron is sitting at between it's sitting at about negative 65 to negative 75 millivolts. Remember, comparing the inside of the cell to the outside. And if a stimulus comes in, we're going to start to see some of the sodium channels open. And then what we see is we get this little bit of a change in charge with charge of the membrane. But not much, and then it kind of dies out. And so what this is showing us here is there was not much of a potential in order for us to activate a response. Oh my gosh, that was a big bump. All right, but we have this magic dotted line up here. This magic dotted line at negative 55 millivolts is very important in generating what we call the action potential. In order for a neuron to fire and make that signal go from the front end, from the dendrite and all the way down to the axon so we can jump that synaptic gap, we have got to reach something that's called a threshold potential. Okay, so that means we've got to activate that neuron enough to get the charge to change to a negative 55 millivolts. If we can get that that charge to change up to the threshold, then all of the sodium gates are going to open and we're going to see some huge changes. So let's see what happens here. All right, because we have not reached the threshold yet, we call this a graded potential. So it's not quite strong enough to get the message to send, and it dies out. All right. Oh, probably made you dizzy there. Sorry. Okay, so the action potentials. This is rapid depolarization. Okay, remember we're getting less and less negative. When a partial depolarization reaches the activation threshold, more of those voltage-gated sodium ion channels are going to open, which then means that more sodium ions are going to rush in. And we are going to change that membrane potential quite a bit. You can see here that it's going to go from negative 70 millivolts to positive 40. That's a huge change, and it's going to happen very quickly. Remember, those sodium ion channels are only open about 1 millisecond. All right, I'm going to flash through here. Okay, and we can see here that when that happens, our membranes, our potentials of the membrane change. All right, ion channels open and close in response to the charges across the membrane. Once the sodium channels have closed, then the potassium channels will open up. Okay, and they are not, oops, I tried to get rid of that picture, sorry. All right, let's look at this graph here. All right, so now we've got a stimulus that comes in, and look at that, it reaches our threshold. Okay, let's see what happens next. Oh my gosh, that's a huge bump. All right, so we've got this, and if we take a look, we've reached our threshold, and so now what we see is we got this huge jump, and look how fast that took place. We jumped all the way up to a positive 40, and remember we're talking about the inside of the membrane potential, and then we drop back down again, and we're going to focus on this first part here. The threshold for all mammals is negative 55 millivolts. That's an important number to remember. Okay. This is the section that we refer to as depolarization, where all the sodium ions are moving in. Okay, and it's due to the electrical and chemical gradients. All right, our change in our amplitude here is 105 millivolts. That is a lot of different sodium ions that are moving into that cell. Okay. All right, so a refractory period here is when the sodium channels are going to deactivate. They don't close. We are going to use something called the ball and chain, and the ball is going to come over. That's like the cotton ball from yesterday. Going to come over and block those channels so that they are no longer in use. And this is going to make it so that the action potential cannot fire again until we have reset the membrane. Okay, so here's kind of a little, which way we go? Open, and then it's closed. All right. So now we talk about something called repolarization. Repolarization is when the sodium channels become blocked because of that ball and chain, and the potassium gates are now going to open because of that change in potential of the membranes, where the outside becomes negative and the inside becomes positive. Okay, that is going to stimulate the sodium ion channels to open up, and it's going to allow sodium ions to move out of the cell, reversing that membrane potential. If you remember from yesterday, the sodium channels were open quite a bit longer, almost 3 times longer than those The potassium channels were open about 3 times longer than the sodium channels were, and so that allows more potassium ions to move out of the cell than any of the sodiums that moved in. And so what does that mean? Hit the right button here. That means that what we see is going to happen is that this membrane potential now is going to repolarize or come back down here to normal. But because it's open so much longer, we end up in a section that drops below where our resting potential is, and this is called hyper, or more than, polarization. So hyperpolarization.